Sawadee-kap, everyone. So I'm Shota Nakamura from Osaka University. So first of all, I'd like to my deep thanks to the organizers and my Thai friends. It's a great honor for me to be here. So today, I'm going to talk about rapid on-site identification of Mycobacteria. So first, let me introduce myself briefly. I'm Shota Nakamura, Associate Professor. So my research field is genomics, metagenomics, microbiome, and bioinformatics and structural biology. So do you know the first NGS technology? This is the 454GS20. So I met this machine in 2006 in Yokohama at Yokohama Riken. So at that time, this machine can determine only 20 megabase per 6 hours. And since then, we have many NGS technology, do you know? So 454 technology already disappeared. So also solid technology also disappeared. But Illumina technology is surviving a long time, right? And also recently, long-read dose PacBio and Minayo nanopower sequencing is rapidly growing. So our institute, Osaka University, has those NGS platforms, Novasec X Plus, Novasec 6000, DNBsec G400, G99, MySeq, Minayon, and P2 Solo. So each sequencer has each performance specificity. So it's important thing is your choice. You have to choose the best one for your application. So this graph shows the data output size of our institute. So it's rapidly growing, and now it reached 350 terabase scale per year. So first, how to use this NGS technology? Our idea is very simple, just to detect pathogens so by using NGS. So extract DNA or RNA from patient and sequence it by NGS. And then data analysts can find genetic information of virus or pathogenic bacteria. So it's like an ideal method for pathogen detection. And this is my old work in 2009. So we detected the influenza virus from nasal swab samples and norovirus from fecal samples, hepatitis C and G virus from blood samples. So we also apply this method to detect pathogenic bacteria, Campylobacter jejuni. So we compare the recovery state of fecal sample and illness sample. We detected Campylobacter jejuni from by metagenomic analysis. So we also did genomic study of monkey parasite, Plasmodium cynomolgi. This one has a close relationship with human malaria, vivax, but far relationship with falciparum. So this is a food poisoning accident in Japan. In 2009, in Osaka, there is a food poisoning, 96 patients. And next year, also we have this food poisoning accident in Tochigi. But pathogenic agent seem to be Clostridium perfringens, but we cannot detect known toxins. So we look for the toxin gene and found a novel toxin, so-called BEC, binary enterotoxin of C. perfringens. And this new toxin has a far relationship with a known toxin like this. So we also compare the many NGS technologies. So this is a comparison of bacterial genomics using 454 life technology, Illumina, MySeq, and PacBio system. So only long-read PacBio sequencing achieved a complete genome of Vibrio parahemolyticus. So in 2014, since then we can determine the complete genome of bacteria. So this is a study of gut flora here. So we compared healthy children and underweight children in Bangladesh. So we found that those children, underweight children has a very different pattern, different flora pattern compared with healthy children. So this is a flora change, flora dynamics during cholera infection. So as you know, cholera infection has a severe diarrhea disease. So in day zero, the before treatment, so Vibrio cholera, this one has occupied half of our flora, our gut flora. But then by taking antibiotics, this rapidly decreased day 2. So such a flora, we are studying such a flora dynamics, studying that flora dynamics of any disease. Sorry. Okay, so this is not infectious disease. This is an ICU patient. If you have a severe injury, so you go to hospital, and then we check their gut flora during this ICU treatment. But as you can see, so patient 1 has a big change, from red color to blue color. So somehow, this host status affected to our gut flora. So rapidly changing such kind of flora, dysbiosis, so-called dysbiosis. So and also we did a research of flora dynamics of COVID-19 patient. We compare severe, mild, and healthy one. And not only bacteria, we compare the fungus flora like this. So somehow severe patient has a lower diversity, lost diversity, compared with a healthy or mild patient. So, okay, introduction finished. So from now, we are going to, I'm going to talk about Mycobacterium diseases. So as you know, Mycobacterium diseases include tuberculosis, leprosy, and NTM diseases. So we have already good vaccine and good treatment for tuberculosis, but good treatment for leprosy, but not for NTM diseases. Because NTM has a wide variety of species, it's over 200 species. So as you know, tuberculosis causes human-to-human transmission, but leprosy is hard to cause human-to-human fish so oftenly. And NTM as well, NTM cannot cause human-to-human transmission. Just the patient take this bacteria from the environment. Sorry. Okay. So that's why risk factor is water environment. So drinking water, hydric soil, acidic soil, and wet land. So somehow the patient get this bacteria from environment. So it's thought to be old female is one risk factor in Japan or developed country. But I heard in Thailand, male is higher incidence compared with female. So this kind of behavior, cleaning bathroom or gardening, is high risk for NTM, thought to be high risk for NTM diseases. And this is a global epidemiology of NTM diseases. So red color showing NTM is increasing. So, but the dark color, gray color shows no data, right? So also whole country, whole world, NTM is increasing now. So this is Japan. Japan's incidence rate for tuberculosis and NTM diseases. So now in Japan, tuberculosis is getting down, but NTM disease is growing rapidly like this. So this is a species list of species ranking of Japanese epidemiology. So top one is Mycobacterium avium, second one is intracellular, and those two account for over 90% of NTM disease in Japan. And then Abscessus kansasii, Gordoni, Fortuitum, Keronide. So like this. And the problem is drug sensitivity. So even these species is not enough for judging drug sensitivity. So those subspecies level, they show the different sensitivity for those drugs. So we need to identify accurately, so at a subspecies level. So do you know this is a classification of biology, so taxonomy from species, genus, family, order, class, phylum, kingdom. So for example, Asia, Japan, Osaka, Suita, Nakamura, Shota, like this, right? So if you use 16S ribosomal RNA technology, we only judge genus level only. So it's like a Nakamura only, right? So this is not enough because, because the culprit is Nakamura in Osaka, but Nakamura is very typical name in Japan. So in Osaka, there are many, over 1,000 Nakamura, So genus level is not enough for identification of NTM. So as you mentioned, the genus level is Mycobacterium. So if you use the DNA hybridization or mass spectrometry, you can identify at the species level, but not reach to subspecies level, right? So it's need to, if you want to identify subspecies level, you have to use more genetic information, seven genetic information or more. So we then used 184 genetic information for identifying NTM diseases. So strategy is very simple. Just extract the DNA from bacterial cells and sequence it by NGS. And then you can get the output read, then mapped to database. And if these output read go to sequencing type 2, the identification result is sequencing type 2. So, but for this method, the database is very important. So we have to establish that we have to make a good database first, and then I get the 16.3 NTM strain from Riken Bioresource Center, and go to culture cell and DNA, and then sequencing it by long read and short read takes a mixed technology, and then we get the complete genome of those strains. And also we collected genome information from public database and 7,411 assemblies from database. And PubMLC is a known database for Mycobacterium, but this one has only 32 species information. So we expand this information to 175 for NTM species, and also 186 for Mycobacterium genus. So we got the expanded DB now, and then we can compare those phylogenetic relationship with like this. So yellow one is a slow growth type of NTM, and green one is a rapid growth type of NTM. So let's try to identify NTM using real clinical samples. So we got those isolated from culture clinical sample from Ryukyu University Hospital. And then this is a non-isolates. We already know the answer. So let's compare this one is by our method. So our method shows the MLST score. So if the y-axis means a score, and the x-axis is isolate. And if you get the higher score, it means accurate identification. So all those 30 samples, we can get the very high score like this. And this is a comparison between mass spectrometry result and MLST burst. So case 1 in the M. caroni, and our result shows M. caroni. But the second one, mass spectrometry says fortuitum, but our method can say subspecies level. M. fortuitum subspecies acetamidoliticum. But the third one shows different results. Mass spectrometry said intracellular, but our method says iongonense. So after careful consideration of genomic analysis, this one, our method is the correct answer. That mass is incorrect one. And also we can get subspecies level of abscessus, subspecies abscessus, subspecies machiliense, also avium subspecies hoministus. So next one is a new trial. Now, let's try to identify unknown isolates. Somehow those culture isolates seem to be mycobacteria, but no conventional method cannot identify species. So let's try identification using our method. And then if you use a conventional database, MLST database, this one has only 32 species information. We got only one clinical sample. Case 11 shows a high MLST score, but other samples shows cannot identify, right? But we now get the new database. We add the Mycobacterium information to 175 species information, and then we can get the very high score for all those 16 clinical samples. So they are Goldoni, Lenchflavum, Wolinski, and Kansaceae, and Europeanum, very rare NTM diseases. So this one shows specificity for our method. If you use 16S sequencing analysis, many species share the high sequencing homology, showing yellow box. But our method uses 184 genes information, so we can get the one by one high score like this, very high specificity. So this is a subspecies level, Abscessus has 3 subspecies, Abscessus boletti and Machiliense. So our method shows only Abscessus has a high score with subspecies Abscessus and also low score with other subspecies. So our method shows high specificity at the subspecies level. So we published this paper in 2019. So and then Japanese newspaper introduced our work like this. So I was very happy. And then next we try to do the rapid identification of NTM. So if we use a nanopore sequencer, the nanopore sequencer can output the sequencing read after input samples. Immediately you can get the output data. So we estimate the time required for identification is only 10 minutes. And then we try to make such a system, for rapid identification of NTM. And the hospital side, just do nanopore sequencing. And the sequencing data go automatically to our institute, and then our facility do data analysis, and then result get back to hospital side, just a text message, like species name and score. So this is a real photo of conducting in Ryukyu University Hospital. So he is actually a medical clinician. So he is inputting the DNA sample to nanopore sequencer here. And then necessary thing is only this one. You need to have a laptop computer and incubator and small centrifuge. So, of course, nanopore sequencer. And the result is like this. You can see species name and score. And this case, we started the sequencing 11:20, right? And the score getting high until noon. So just it takes 40 minutes. And the result is Mycobacterium abscessus boletti. And also you can put multiple sample to one sequencing run. So barcode 3 shows same subspecies, some species information, abscessus boletti. So I'd like to introduce a real clinical case for NTM Japan. So case 1 is a female, 44 years old. And she was transferred to Toneyama Hospital due to repeatedly respiratory tract infection. So despite medical treatment with erythromycin, patient symptoms become worse, and unidentified rapid growing NTM were detected in her sputum. So we did, Toneyama Hospital has our system, so they tried to identify this NTM using our system, but score is not so high, 0.643. So it means our database has no information about this species. Then we did a complete genomic sequencing of this species. And then I found this species has a close relationship with Mucogenicum, but their genomic similarity is very low. So this one is ANI index. This one shows genome similarity. If this value lower than 95%, this means a different species. So this one, the most close one has 93.3%. Now this one lower than 95%. Then this means a new species. So we reported this new species, and we named this one as Toneyama churis. So because hospital name, we took a hospital name to this species, new species. And also case 2, 74 years old male. This patient is admitted due to the fever and dyspnea. So this chest CT shows a significant shadow of left upper lobe. And also unidentified NTM were detected in his sputum. So, so then it's the same thing. So this non-NTM has a close relationship to M. vulneris, but showing 93.4% of any index. So it means a new species. And we judged it as a new species and named it Mycobacterium sendiense. Sendi is a town name of Osaka. So patient address is Sendi, so we named it as sendiense. So case 3 is a 59 years old female, and chest CT shows bronchiotaxis like this. During follow-up, sputum cough appeared, and the treatment with erythromycin was started. At once, M. caroni was detected, but disappeared after 2 years. As the disease progressed slowly, so again, unidentified rapid growing NTM were detected. So this one has, we are very surprised. This one is not Mycobacterium, different genus. This isolate belongs to the genus Tskamurella. So Tskamurella comes from famous Japanese scientist Tsukamura sensei. And Tskamurella and shows 86.2% ANI index to close 1. So we judged it as a new species and named it Toyonaka-ense. Toyonaka is also a town name of Osaka. So this is a pulmonary disease caused by Tskamurella Toyonaka-ense we reported. And case 4 is an 82-year-old female diagnosed as a Mycobacterium gordonii pulmonary disease. So keeping clarithromycin monotherapy for 15 years, very long time, but switched to erythromycin monotherapy due to gastrointestinal symptoms. And she developed this effusion and underwent thoracic drainage. But her symptoms gradually worsened. And then this one is, conventional method says Mycobacterium gordonii, but this one, the Malditoff, this is a commercial service of species identification. So Mass said this is gordonii, but our method said this one is not gordonii, different clade. So that's why we reported it is a new species, and there are two novel species related to this symptom, and Mycobacterium novus gordonii and Mycobacterium shin gordonii. So do you know shin gordonii? Shin, what shin means? It's in Japan. In Japanese, this shin means new gordonii. So I take this word from Godzilla. Do you know Shin Godzilla? So this is a new Godzilla, the new monster, right? So now, so many new NTM monsters are attacking to Osaka. But do you think this is a really new monster or no? So I think this is not a new monster. Actually, we don't know, So true diversity of whole mycobacterial species. So maybe just the tip of iceberg is a clinical isolate, Avium intracellular. But in nature, in natural world, there are more diversity. Mycobacteria may have more diversity, like Torreya maturis, Senriense, Toyonakaense, and Shin gordonii. So next, anyway, we can do rapid identification in hospital using nanopore and cloud computing system. But is it really rapid? Or, I think no. So because we have to use culture. So this culture takes a very long time, right? So first culture, midget culture takes 2 or 4 weeks. And also second culture, agar plate takes further 2 or 4 weeks, right? So we want to make this protocols more short, And then we try to use early stage of midget culture, liquid culture. Then 3 or 5 days, just to take the bacterial cell from liquid culture, and then do nanopore sequencing and drug resistance prediction. So important thing is key drug clarithromycin, resistance or sensitive. So subspecies Abscessus and Boletti shows clarithromycin resistance, but Masliense showing sensitivity for clarithromycin. And then, but this is only species name, right? So we have to see a real cause of drug resistance. It's 23 ribosomal RNA point mutation. So if you can see this point mutation at ribosomal RNA gene, this means drug resistance. And people often say the nanopore is low accuracy, but I believe it's enough accuracy for judging point mutation already. So Minayon version R10.3 has a 100% accuracy for those target sequencing lengths. So we judge that this one is, we can see the point mutation using nanopore. And also, we also look at the ARM41 gene. If you find truncation of this gene, this means sensitive. But if you have a full length of this gene, this means drug resistance. And this one, for drug resistance prediction, we use 2 genes, 23 ribosomal RNA and ARM41, the truncated or full version. So this one is a real clinical case of 45 NTM diseases. And we compare the PCR test and our method, core genomic MLST. And in this case, the case 6 showed we found a point mutation of ribosome RNA A from T. And this seemed to be drug resistance. And the real test showing clarithromycin resistance. And also case 11 shows intracellular subspecies chimera. This one has a point mutation and resistance. And intracellular also has AC mutation, resistance, and all those samples match to real test of drug resistance. And please see this one, 39 samples is showing subspecies abscessus. And this one has a complete ARM41 gene, and this one means drug resistance. And real test shows same results. And case 40 is Machiliense, but ARM41 is truncated form. So this one is sensitive. So every hour prediction is matched to the real test of NTM test. So we reported to this MIGIT-seq, you can do identification from MIGIT samples, and then we publish this paper. So next is, okay, so we now have, MIGIT-seq can identify NTM and predict drug resistance from 3 or 5 days from clinical samples. But it's not so rapid, right? We have to wait 3 or 5 days. So next, we try to make this protocol more faster. Sorry. And still, it takes MIGIT-seq take 3 or 5 days. So if cut the culture steps, just take a sputum samples and then sub-treatment and NALC NaOH treatment remove other bacteria. And then try to get the Mycobacterium information from this sample. So to do this, we use the target capture sequencing for Mycobacteria. So do you know that exome sequencing or some targeted sequencing use this technology? Just take a target sequence only, target DNA only by using magnetic beads. And then we already have a database for NTM. So we designed many probes, many target capture probes for NTM. So again, I explain target capture. Just we know, we necessarily think it's a target sequence showing Bazenda. So take this one by using magnetic power, so get the target gene only and remove other genetic information. Then this one is a result of NALC-seq, because this one can identify NTM from NALC treatment. And this one, y-axis shows score of MLST. And those sample set classified into smear positive, smear level, smear plus 1 plus or 2 plus or 3 plus. And then in total, we get 98.1% sensitivity. But for smear positive, over 1 plus, we can get 100% sensitivity and 100% accuracy. So I already explained conventional test takes first culture and second culture, and it takes maximum 8 weeks getting drug resistance information and species name, right? But MIGIT-seq can shorten that second culture. Just you use first culture samples, and then usually 2 weeks, maximum is 4 weeks. And NALC-seq can skip this first culture, and this one can identify NTM within 24 hours. So this is a real turnaround time we measured so identification. So conventional test takes 28.5 days for identification, but the MIGIT-seq can do this within 70.3 days. And the NALC-seq, very short, can get results within short time, in average 90 hours. So we published this paper just last month. So please see this on the 2025 May 21st, and we are very happy to have this information with you. And now we have this website, so MSD Bus, so you can use our system freely. So just create an account and upload your data to the website. So you can get the species information and drug prediction. So 2 days ago, they used our system like this. And Omu-san and Poi-san is now look for the data and upload this one to our system. And this one is Yok-san is seeing our result. So this one is a real result from their data. So actually, this one is number one is Mycobacterium fortuitum. So we already get a user from Thailand. So please use this system and please accelerate the identification of NTM here. Maybe you may have a wide variety of NTM in Thailand. So please look for new species in Thailand. Together with me and my collaborators. So now you now have two options, MIGIT-seq and NALC-seq. And MLSD Bus Web system. Our system will help you to identify NTM from clinical samples within a day. Right? And without bioinformatics, it's very important. Bioinformatics is a complicated process, right? So just upload data and then get the results. It's easy. So that's all. Thank you for your kind attention. ขอบคุณมากครับ
ขอเสียงปรบมือครับขอบพระคุณนะครับดร.โซตะ นากามูระครับโดโมอาริกาโตโกไซมัส